So I'd just like to introduce Stephen Montgomery, who works on brains. So he started with very large brains and of cetaceans and primates, as he's going to talk to us about today, and then has moved down the scale and is now peering into very powerful microscopes and looking at butterfly brains. But I think we're not going to get too much of that. His academic career, though, has sort of gone in the opposite direction, at least as far as academic institution is concerned. So he started at Cambridge with an undergraduate degree there and then a PhD. He then moved to Oxford and did a junior research fellowship there. And finally, he's moved, well, I don't think finally for his career, but at last, should I say, he's moved to UCL and is now a Leverkusen research fellow in the genetics department. And today, we're going to have a talk about primate brain expansion. Thanks.
Okay, thanks very much, Kit. Thank you very much for the invitation, especially since you've heard it all before. But, yeah, so I'm going to talk about some work I've been doing. Most of it is from my PhD work. But some of it is sort of stuff that's dragged on a bit since then. But the starting point, which I don't need to tell a room full of anthropologists, is that primates are a very diverse group of mammals. So if you consider them just in terms of size, they vary from less than 50 grams to 150 kilograms. They have a range of dietary adaptations, frugivory, folivory, and some nice examples of tool use for extractive foraging. They have a whole range of sensory adaptations. You have nocturnal primates, diurnal primates, and some with very specialized sensory ecologies like the aye-ayes that have this massive ear so that they can hear grubs in branches that they tap with their fingers. And of course, Kit's favorites, you have a whole range of social systems, from the complex vision fusion systems you see in chimpanzees to monogamous pairing and smaller group sizes in things like tiddy monkeys. And this diversity is represented or is present in measures of brain size as well. So if you look at any of the major groups of primates, strepsirrhines, neuromonkeys, monkeys, or apes, there's quite a big range of brain masses. And in each case, the range overlaps between different groups. And if you look at brain size relative to body size, for example, using Jerrison's encephalization quotient, then again you see a huge range in all the major groups. And it's generally thought, and there's good evidence, that this variation in brain size has some sort of evolutionary significance. There's quite a big debate over how best to measure brain size, whether or not you should think about EQ or relative brain mass, or maybe there's a school of thought that total brain mass, or maybe the total number of neurons in the brain, is the most pertinent trait. But for each way of measuring the brain, there's evidence that there's an association with visual ecology or social ecology, or some measure of general cognitive ability. And there's another idea that maybe offers some sort of behavioral flexibility or a cognitive buffer that allows primates to respond to variable environments. We also know that brain structure itself can be shaped by selection pressures. So in the brain, there's a whole group of different components. And some nice work by Rob Barton and Paul Harvey showed that functionally related components in the brain co-evolve with each other, but regions of the brain that don't directly function with one another can evolve independently. So selection can act on specific parts of the brain. And you see this in terms of the ecological factors that vary with different regions of the brain. So, for example, social group size is known to correlate with neocortex size. You have visual brain volumes correlating with various aspects of stereotypic convergence and activity pattern, olfaction with diet and activity pattern again, and the cerebellum size with extractive foraging. And it's this diversity in brain size and structure that evolutionary biologists who are interested in the brain are trying to explain. And the work I've been doing has mostly taken a genetic perspective, and you might ask why you would choose to do this. And one reason you get quite a lot is that it's put to you that there is a distinction between processes above the level of the individual, so how an individual interacts with its environment and how that relates to fitness, relating to ultimate evolutionary questions, so why this thing evolved, or why it happens that way. Whereas processes below the level of the individual in terms of their genetics and development are more about proximate questions, so how things develop. But there are also lots of ultimate questions that you can ask about these cellular genetic developmental mechanisms. So the questions that I'm interested in is whether or not independent changes in brain size share a common genetic basis. And if they do, then why are particular genes or developmental pathways targeted more often than others? Does this reflect some sort of genetic or developmental constraint that channels brain evolution in a particular way? And is the coevolution that you see between different brain structures or between brain and body size due to genetic constraints that mean that that's the only way that those traits can evolve? And finally, a big question that anthropologists are interested in is how species-specific genetic changes relate to species-specific phenotypic changes. Okay, so when you think about the genetics of any phenotypic change, you can study different categories of genetic change. So if you imagine a gene is a sequence of DNA, and part of that gene will encode a protein. Another part of that gene will encode a regulatory element that controls how that gene is expressed, so whether or not it's turned on or off, and how much of the gene is produced. But when it is turned on, this gene is transcribed into RNA and then translated into a protein, and this protein goes off and does whatever function it has in development of the organism. So you can think of different ways that this schema can change. You can change the protein coding gene itself. This will change the structure of the protein and potentially the way the protein functions. Alternatively, you can change the regulatory element either by changing the amount of protein that's produced, so upregulating it or downregulating it, or potentially duplicating the regulatory element so that you have multiple regulatory elements that can then take on functions in different tissues. For example, expressing at one level in the brain and another level in the spleen or the liver or whatever. And I'm mostly going to be talking about this sort of evolution, so protein coding gene evolution. And this is mostly for practical purposes. If you want to compare the protein coding genes of different species, all you really need is a bit of genomic DNA that you can amplify gene sequences from. You don't have to control for the environment or the age because the DNA is always the same. If you want to look at gene expression levels by sequencing the mRNA to see how gene expression varies across different species, then you have to control for the environment, you have to control for age and various other things, so it becomes very difficult, especially in primates where you're not able to sort of harvest tissue whenever you want. So this is a very brief introduction into how protein coding genes evolve. And the starting point is to recognize that the amino acid code is degenerate. So that means a gene sequence is composed of codons of 3 nucleotides. And these codons can have 4 possible bases, A, T, G, or C, or an RNA will be represented as A, U, G, or C. And there are more possible combinations of these 4 bases than there are possible amino acids, which means that some changes in the nucleotide sequence won't change the amino acid that's incorporated into that protein. So, for example, if you change UCC to a UCU, you still get the same amino acid. So this sort of change is called a synonymous change. And because it doesn't affect the protein sequence, it's effectively neutral to selection. So it won't have a fitness effect, basically. So the chances that that nucleotide change will be fixed is basically determined by drift. Alternatively, some nucleotide changes do change the amino acid that's incorporated into the protein. For example, if I were to change UUC to UCC, you'd get a different amino acid. So this is a non-synonymous change, and it will affect the protein that's produced, and potentially that will affect the function of the protein. So this is therefore non-neutral with respect to selection. And that means it will be fixed according to how it affects the organism's fitness. So you can compare the rates at which these two types of mutation are fixed in a population by looking at their rates. So you calculate something called the DNDS ratio, which is just the ratio of non-synonymous substitutions to synonymous substitutions. And if you have a ratio of 1, it tells you that the rate at which these mutations are being fixed is the same as these mutations. And that tells you that the gene is evolving neutrally, because it implies that these non-synonymous substitutions are equally likely to be fixed as these synonymous substitutions that are being fixed by drift. If, on the other hand, and this is for the majority of genes, DNDS is below 1, it tells you that there is some action of selection to remove non-synonymous chains from the gene pool. And that is probably because they have a deleterious effect on fitness. And finally, in some cases, you'll have a DNDS above 1. This tells you a mutation has occurred that changes the non-synonymous, there's a non-synonymous change that changes the protein's function, and that function, that new function is being selected for by positive selection. So these types of mutations will then be fixed at a higher rate than the synonymous rate. So this basically gives you a rough idea of what sort of selections a gene or a part of the gene is under. So to do this sort of analysis, what you need is an alignment of the same gene from different species. You need to know how those species are related to each other, and you need a model of evolution that's based on that explanation I gave you. And with this, you can sort of compare the selection pressures that are acting in different parts of the phylogeny and infer changes in the rate of evolution, identify episodes of adaptive evolution, and potential shifts in the selective regime acting on a gene. But what it doesn't tell you about is whether or not these changes in the DNDS ratio have any relevance for a particular phenotype. So to make that link between selection on a gene and changes in a phenotype, you need some sort of phenotypic framework that helps you interpret your genetic results, and you also need to perform explicit quantitative tests of gene-phenotype coevolution. And this is what I've basically been trying to do. So the first part I'm going to talk about is developing a phenotypic framework for interpreting my molecular evolution. So to do this, I assembled a data set of brain mass and body mass across living primates, and including some fossil species where they're available. And with this phylogeny and data, what we can do is we can apply different models of evolution to the tree and test whether or not one model fits the pattern of phenotypic evolution more than another model. And most of the results I'm going to talk about were performed in a Bayesian context in this program called Bayes traits. So one of the first things we're interested in is looking at macroevolutionary trends. So a macroevolutionary trend is where phenotypic evolution is more likely to proceed in one direction than another. So to test this idea, what you can do is you can compare a model where phenotypic evolution moves at random to a model where there's a directional bias. And these plots show the posterior distribution of likelihoods, or how well each model fits a phenotype, for a non-directional model in blue and a directional model in red, for body mass, brain mass, and relative brain size. So what you can see for body mass is that the two models fit the data more or less equally well. And this tells you that you can support a directional model, so you infer that there's been a random pattern of evolution for body size across primates. So it's equally likely to go up and down as you move across the tree. In brain mass, however, there's strong support for a directional model, and this means that during primate evolution there's been a tendency for brain mass to increase in size rather than to decrease in size. And these two differences between brain and body mass mean that when you look at relative brain size, there's very strong support for a directional model. So there's very strong support for the idea that brain expansion has occurred across primates, more or less throughout their whole evolution. Another way of looking at that is to plot the amount of phenotypic change for each trait across your phylogeny. So this is just the relationships of the living species that I showed you before, but in this case, the branch length is proportional to the amount of phenotypic change that occurred on that branch. So this is for brain mass, and you can see that the ancestral primate had a very small brain, and early on in primate brain evolution there's been these big increases in brain mass, and more or less the whole tree is dominated by positive increases in brain size. And there's only really a few examples here in the callitrichids and here in the cherogalids where brain mass has decreased. And overall, brain mass spent 94% of evolutionary time during primates getting bigger, whereas body mass increased only about 50% of the time. And you can see that again when you look at relative brain size, all the branches are moving this way across the slide, which tells you that they're all increasing in size as you progress from the last common ancestor of primates through to the living species. And there's only really a few small blips in this pattern where relative brain size decreases. The most notable one is in gorilla, and that decrease is caused by a really big increase in body mass. So this is another way of looking at the same data. This time what we've got is time along the x-axis, going from the last common ancestor of primates through to the present day. And in red you have brain mass in log milligrams, and blue is body mass in log grams. And this is just plotting it as you go through from the ancestral primate through the tree to each species tip. So in this case this is humans and chimpanzees, so this split is humans going up and chimpanzees here. This is spider monkeys and the eye, and basically what you can see is that in these independent lineages there have been parallel expansions in brain mass. In these two examples you have the marmoset and the mouse lemur, the same thing, but this time you can see at the origin of callitrichids, body mass has taken a nosedive and started decreasing, and brain mass has sort of trundled along, decreasing slightly. And you see the same thing in mouse lemurs. There's a big decrease in body mass, whereas brain mass decreases much less. So this just tells you that brain mass and body mass are increasing independently in different lineages, and in some cases decreasing. So the results from this work sort of give you an idea of how brain size has evolved across primates. And the main things to take from this for the genetic stuff can be broken down into these four categories. First of all, if you look at brain and body evolution, they have different characteristics. This is quite important because it suggests that brain mass can evolve independently of body mass. Then we can look at the directionality and what does this strong pervasive parallelism in brain mass mean. Basically it tells us that there's been strong selection for increases in brain size across primates. This has occurred in all the major lineages of primates, and it's not just limited to the lineages leading to humans. And there are only really a few small episodes of brain mass reduction. Okay. So that tells us that brains have gotten bigger multiple times. Now we're going to move on to talking about how selection might act to build a bigger brain. So the basis of our initial studies, we're trying to identify genes that might have important roles in development. And we focused on a model of cortical development proposed by Rakic, which is quite popular. And basically his idea was that the cortex is composed of these columns of neurons, where each neuron within a column is descended from the same ancestral cell lineage. And what he proposes is that at the beginning of brain development you have a pool of progenitor cells or stem cells, and these cells self-replicate multiple times, and as they do that you have an exponential increase in the number of these progenitor cells And eventually they stop dividing in this replicative way, and they produce another type of cell called a radial glial cell. And this cell divides asymmetrically. So it will produce a neuron that will travel up to the cortical plate, and then it will also produce another copy of itself. The next round of division, it produces another neuron that goes and sits on top of the neuron it produced last. And so it goes through these successive rounds, producing neurons and forming these cortical columns. And based on this model, you can sort of identify two key switches in development. One is the switch going from these symmetric divisions to these asymmetric divisions. And then the second switch is going from these asymmetric divisions to the terminal division, where that cell stops dividing. And these switches are quite important. If you change, for example, the duration of these symmetric divisions, you very rapidly increase the number of neurons in the cortex. So this is just adding one extra symmetric division of the progenitor cell, and you already double the number of radial units that you have in the brain. Alternatively, you can change the amount of times these radial glial cells self-divide, and this will determine how many neurons are in each radial unit. So you have sort of two axes of potential variation, one in changing the number of radial units, and one in changing the number of neurons in each radial unit. And both of these cell fate switches are determined by the way the cell divides. And it's quite a simple idea, really, that it's the cleavage plane at which the cell divides that determines the cell fate. So the cleavage plane is just, if you imagine a cell as a big sort of balloon, if you pull a piece of string around it to make two balloons, it's the way in which the angle at which that piece of string pulls the middle of the cell together. So during symmetric proliferative divisions, for example, this division plane is vertical, so this cell is going to divide by doing that, basically. And this means that this surface membrane at the bottom of the cell is inherited equally between the two cells, and this is where there are probably some sort of morphogens that determine what cell the daughter cells develop into. So if this is inherited equally, then you have two similar daughter cells. In this case, the division is asymmetric, so one daughter cell inherits all of this morphogen, and the other daughter cell doesn't inherit anything, and that will produce differentiated daughter cells. So in this case, self-replicating once and producing a neuron. So we're interested in identifying genes that have this sort of function and might be able to influence these genetic switches, and I'm going to talk about some of them now. In this case, we're going to talk about the microcephaly genes, and in this case, we're going to talk about a single gene called MIN. So microcephaly is a pretty horrible disorder that specifically affects brain development. So it's associated with a reduction in brain size, about 4 standard deviations away from the population mean. So this is a microcephalic individual on the left, and this is a typically developed individual on the right. And you can very quickly see that it's sort of messed up the development of brain size. And this is, as you would expect, associated with behavioral problems, mental retardation, but there's no other neurological deficit, and there's no other defect in development, for example, with body size or whatever. And these genes are linked to the proliferation of progenitor pools that are undergoing symmetric divisions. So previous studies have looked at the molecular evolution of these genes. So again, this is using these DNDS ratios that I talked about at the beginning. And they're sort of comparing mostly apes with a couple of monkeys. And the key result that they argued is that you have these high numbers of DNDS along the lineage leading to humans. So in a few cases, they're above 1. That infers that they're under positive selection. But in general, they're making this argument that these branches in red are accelerated relative to the rest of the tree. And people have looked at a few different genes associated with this disorder, and what they tend to find is that on some deeper internal branches, there are sort of high rates of evolution, but also on terminal tips, there's high rates of evolution that are not limited to humans specifically. So the claim was made that these genes have a function in brain evolution, but really they left a few questions outstanding. First of all, it's not clear whether or not the positive selection was restricted to the human lineage, and if it does, we don't have any direct evidence that this is because they're under selection to produce changes in brain evolution. And it's possible that they contribute to other traits. For example, they're known to have functions in spermatogenesis, and genes associated with sperm development and production are frequently under positive selection due to mate conflicts. So we tried to address these questions by collecting gene sequences for these loci across a broader range of primates that represent different episodes of brain evolution. So this is our data set for the apes, 6 older monkeys and 10 or so newer monkeys. This represents a variation in brain mass and variation in relative brain size, and you can see that in each of the clades you have variation from large brain species to smaller brain species. And the first thing we wanted to know was whether or not you can see a signature of evolution across the whole tree. So this tells us that these genes are targeted by positive selection throughout, in this case, anthropoid evolution. And this was done with a test that looks for positive selection at subsets of the gene. But basically, the results tell you that, yes, all 4 of these genes are evolving adaptively across primates. And you can do various tests to see whether or not this positive selection is limited to different parts of the tree. And in each case, it doesn't seem to be. So it's not the case that these genes are only under positive selection during human evolution. But we still haven't explained what this positive selection is trying to produce. So we wanted to explicitly test some of our hypotheses. And the overriding hypothesis here is that microcephaly genes contribute to the evolution of neuron number and therefore should co-evolve with brain mass. And we developed a couple of hypotheses based on some features of neurogenesis. First of all, we know that brain mass correlates with total neuron number better than relative brain size. And second of all, we know that neurogenesis occurs prenatally. So any infant primate will be born with all the neurons that they have for the rest of their life. So that means that postnatal brain growth doesn't reflect changes in neuron number, it reflects sort of synaptic growth and axonogenesis and things like that. So based on these two facts, or whatever you want to call them, can come up with some testable hypotheses. First of all, we'd expect that if microcephaly genes are co-evolving with neuron number, they should be more strongly correlated with absolute brain mass than relative brain size. And second of all, because this postnatal brain growth introduces more sort of noise to any sort of co-evolutionary pattern, we would expect there to be a stronger correlation between microcephaly gene evolution and neonatal brain size rather than adult brain size. And overall, the most relative phenotype is absolute neonatal brain mass. And the way we test this is to basically calculate the average DNDS ratio from the last common ancestor of the species in our data set to the species tip. So for example, for the capuchin, we calculate the DNDS along this red branch. We do this for each species, and this produces a measure of selection that is comparable with species data on brain mass. And then we're basically performing a correlation analysis between this measure of selection, DNDS, and brain size across the tree. And this analysis corrects for the non-independence of this data. And we can do this with different phenotypes and use something called Rakic's information criteria to compare how well different phenotypes fit the molecular data. So when we did this, we found 2 genes that have a positive correlation between DNDS and brain mass, ASPM and CDK5-RAP2. And when you compare different phenotypes, these are the AIC comparisons, In both cases, you have a lower AIC for neonatal brain mass than you do for either adult brain mass or neocortex volume, which suggests that our hypothesis that these genes should be closely related to neonatal brain mass is sort of validated. There is one interesting outlier for ASPM at this point is the common marmoset, and we were sort of interested to know why this species is an outlier to the general pattern across primates. And in general, marmosets are pretty interesting because, as I showed you during the phenotypic analysis, there was this major reduction in body mass during callitrichid evolution. So we looked at this in a bit more detail by reconstructing patterns of body mass evolution across callitrichids. So these are the tamarins and marmosets and Geldes monkeys. And what we found that basically across the whole group there's been these major parallel reductions in body mass. So this is adult body mass and neonatal body mass, the same thing. So you read down the phenotype on this x-axis, and as you're moving across here, you're basically showing that during evolution along this branch, there was a decrease in body mass. And you can see that for adult body mass and neonatal body mass, the pattern is very similar. And this basically tells us that the major reduction in body growth occurred prenatally. And because brain development occurs prenatally, this slowdown in prenatal body growth would also have effects for other developmental processes like brain size. And sure enough, when you look at patterns of brain evolution within the callitrichids, they show this major decrease too. There is one notable case. The pygmy marmoset here has, on this terminal branch alone, which is only about 1.5 million years in length, there was a 2/3 reduction in body mass. This is the highest rate of body mass evolution that we've identified in primates. And basically, it's probably because this species is some sort of neotenous dwarf, but it's not really known how this major reduction in body mass was brought about. Anyway, that gives us a hypothesis for why the marmoset is an outlier to the general primate pattern, specifically because it had this decrease in brain mass. So we wanted to test this again. We collected some more data across callitrichids and did the same test. So again, there's positive selection just within this subset of data. So ASPM is still evolving adaptively in callitrichids. But this time when you do the phenotypic gene phenotype coevolution test, you find a negative correlation. And that fits in with this idea that ASPM is controlling the evolution of brain size, and in callitrichids brain size has been getting smaller. So this sort of suggests that ASPM is associated with increases in brain mass across most anthropoids, but in callitrichids it's associated with decreases in brain mass. You'll notice this outlier, which is the pygmy marmoset. So again, we don't really know what that species is doing. Okay, so the main points from this section of the talk is that these key developmental genes that have been linked to human brain evolution show patterns of positive selection across primates. 2 of these genes show a consistent pattern of coevolution with brain mass, and for ASPM, this association is present when you're looking at species where brain mass has increased and when brain mass has decreased. And in all cases, the human data point fits the general primate pattern exactly like any other species. So I'm going to quickly talk about this other gene. So this is a gene potentially controlling the number of neurons in each radial unit. So I'm not going to go into it too much, it's just to quickly show that there are other aspects of this diversity. But nenine is basically another protein that changes the way in which cells divide. And this figure basically shows that if you knock it out, then you have a reduction in the number of neurons in the cortical plate. And we know that there is variation in this aspect of primate brain size, because if you look across different species, there's no correlation between cortical mass and the density of neurons per given unit area of cortex. So we went out and collected data for some more primates again. did tests for positive selection, found more evidence of positive selection, and then repeated these gene phenotype association tests. This time, the picture is more complicated. So within catarrhines and within platyrhines, there's this positive correlation, as you would predict if these genes are involved in brain evolution. But in both cases, there's an outlier. In this case, humans, and in platyrhines, it's the capuchin monkey. I could tell you a long story about why that could be, but I'm not going to. But the major pattern you can see is that there's this shift between the two groups. And this sort of fits with the data that exists that suggests that platyrhines have a higher density of neurons per unit area of cortex. And we can test this directly using this data. So here we would expect that the selection pressure acting on ninine would correlate with the number of neurons under a given unit of area, which is our correlative for the number of neurons in a radial unit. And you can see that there's this pattern of coevolution, but because there isn't very much data for the neuron numbers, then it's not quite significant. I won't see that very quickly because it's not important. The main point is to say that these results suggest that brain evolution may have a partially conserved genetic basis. But the next big question is whether or not these genes are sort of part of a small key set of genes that have this role, or whether or not you have a more widespread pattern of genetic conservation across species. And this is potentially important if you're interested in lineage-specific patterns of evolution. So if you want to understand why humans are different to other primates, for example. And I'm going to try and explain why using these Venn diagrams. So if you imagine three independent changes in brain size, each one of these circles represents a different change in brain size. Potentially you could have the majority of genes involved in each independent episode of brain expansion being shared across these three groups. Alternatively, you could have a pattern where none of the same genes are shared across these three independent episodes of brain expansion. Or you could have a situation where normally it's the same set of genes, but in one particular species, that has sort of gone off the beaten track and has something special about them. And typically when you read papers about human adaptation, human genomics, this is the sort of implicit hypothesis that they have in mind, where humans are genetically differentiated from other primates in terms of the mechanisms that underlie phenotypic evolution. But this hasn't really been tested, so we don't know whether or not this is true. It could be that this is the case, so all primates are sort of as genetically differentiated as each other. Or it could be the case that the majority of genes involved in human brain evolution are also involved in, for example, capuchin brain evolution. So most of the stuff that's out there in terms of primate genomics is looking for positive selection on the human lineage. And so this is sort of going from a candidate gene approach that I was talking about before to taking all the genes in the genome and comparing them with another species or a group of other species. And because they were the first genomes to come out, most of these studies compare the human branch, the human genome to the chimpanzee genome. And they're looking to try and identify episodes of positive selection on the human branch. And there are a whole suite of studies have done this, and you get sort of interesting things to do with speech or hair or hearing or brain development. But now there's increasing amounts of genomic data. So this is just an image pulled from the Ensemble Genome site. And this was only if the human genome came out in 2004. And now, 10 years later, there are 43 mammal genomes, 13 of which are primates. So there's now enough data to do more sort of complex pattern tests of adaptive evolution in primates, and to test these hypotheses about whether or not humans are in some way different from other primates. And there is some evidence that there is more widespread convergence in the patterns of selection shaping the genome than may be recognized by most studies. So this is an analysis done for the gorilla genome that shows sort of the amount of parallelism in selection pressures between either the gorilla and chimpanzee in red, the gorilla and human in green, or the human and chimpanzee in blue. So this is sort of quantifying the proportion of the genes, proportion of the genome where you have positive selection in humans and the same gene positive selection in gorilla or some other pair. And along the x-axis are just if you look at different statistical cutoffs, but the main pattern is this is the sort of standard cutoff. If you look at patterns of selection within any pair, you see strong evidence of parallelism, particularly between humans and gorillas or humans and chimpanzees. Interestingly, there's not a lot of convergence between chimpanzees and gorillas. And a nice example of why this is important comes from early studies that have been trying to identify human-specific adaptations. So in 2003, one of the first studies comparing the human genome with the chimpanzee genome found that genes with high rates of evolution in humans are enriched for proteins that have roles in hearing. So you can make a nice inference that hearing is important in humans because we speak to each other. But based on the analysis of the gorilla genome, if you take the genes that have parallel acceleration of evolutionary rate in humans and gorillas, you find that they are also enriched for auditory genes. So positive selection has acted on these auditory genes in humans and in gorillas independently. So based on that, you wouldn't say that they're associated with language. So we've, together with Amy Bodie from Derek Wildman's lab, we tried to come up with a way of testing these hypotheses at a large genomic-wide scale by taking 3 pairs of primates that represent independent episodes of brain expansion. So in one pair we have human, we're comparing it to the orangutan. We have another pair, the baboon, comparing to a colobus monkey, and in newer monkeys, a capuchin and a squirrel monkey. And these are these diagrams that show the amount of phenotypic change on each branch. We're mostly interested in brain mass, and you can see there's sort of nicely equal amounts of phenotypic divergence between each pair. So what we did is we took these 6 species, found all the protein coding genes that we could that existed in all 6 species, and then we did a bunch of evolutionary analyses. So the first thing we did was to do what's normally done in human genome scans, which is to compare each gene individually, looking at whether or not it's evolving under positive selection in humans compared to orangutans. And we did this in each pair, basically looking for an acceleration of positive selection. And what we found is that you get the same number of genes under positive selection in each big brain lineage, but there's no overlap in what these genes are. But when you look at what these genes are actually doing, you do find that they're the same sort of processes involved. So one other way of looking at this was to take these 3 branches, lump them together so we have a big brain branch set, and compare it to these other 3 branches, so a small brain branch set, and look for positive selection across those 3 branches. So if you take these 3 branches, the big brain set, what we find is an enrichment for genes that are involved in something called glutamate receptor binding. And these genes seem to be linked to the functioning of receptor binding in synapses. And these sort of categories of genes have previously been linked to synaptic plasticity and are associated with things like autism and bipolar. So this suggests that there's some level of convergence between some level of conservation in the genetic basis of these independent episodes of brain expansion. Another way of looking at that is to do the quantitative gene phenotype test that I talked about with microcephaly genes, where you calculate the DNDS ratio from the common ancestor to each tip. You do this for all the species and then do phylogenetic gene phenotype correlation tests. When we did this, we found that about 10% of the genes that we were able to look at are significantly correlated with brain mass independently of body mass. And when you look at what these sorts of genes are, you find sort of the sorts of genes, gene functions that you'd expect. So the genes that encode proteins involved in neuron outgrowth, neuron transmitters, and neurodevelopmental disorders. So this again suggests that potentially quite a large proportion of the genome has a conserved role in phenotypic evolution across these 6 species. But I'm sure you will ask whether or not these are particularly robust because you only have 6 data points. So what we did was we took some more data that was available, and we were able to retest about half our data set with a larger taxon size of 13 species. What we found is that you can reproduce these significant correlations for about a quarter of our genes. So this is showing a couple of examples. This is a gene called GRIN3A, which is another glutamate gene associated with autism. This is the pattern across 6 species, and this is the pattern across 13 species. This is another gene, this time a neurotransmitter that's associated with autism again, across 6 species, and then across 13 species. There are some other interesting patterns. So, for example, these data points I've circled here are both callitrichids. So they're sort of showing a similar pattern of outlining the general trend that we observed for ASPM. But the general point that we wanted to get across is that if you do this sort of phenotypically orientated genome scans, you see some evidence of this pattern of evolution, where there's a conserved genetic basis of independent episodes of brain expansion. But you see very little evidence for this sort of species-specific exceptionalism. So this suggests that some genes, potentially quite a lot of genes, have a consistent role in brain evolution. And we can't find any evidence that the sorts of changes that have occurred on the human branch are different to the sorts of changes that have occurred during other episodes of brain expansion. So very quickly, the last thing I was going to talk about is looking at other mammals, so moving beyond the diversity of brain sizes you see in primates to talk about some other aspects of brain evolution. And in mammals, there's this thing that Paul Monger has called the over 700 gram club, which includes humans, seals, elephants, and some cetaceans that all have brain masses over 700 grams. And he decides that 700 grams is an important barrier to get across. But we've also been looking at some patterns of evolution of some members of the over 700 gram club, specifically cetaceans. So doing the same sort of phenotypic framework analysis that I talked about at the beginning, this time looking at patterns of evolution across toothed whales and baleen whales. And you see some interesting differences. So this is a scatter plot of the brain body mass and EQ data for cetaceans. These are the same as the directional model results I showed you for primates. And basically what you find in cetaceans is there's a strong directional trend for body mass and brain mass, but not for EQ. So this tells us that the pattern of macroevolutionary trends in cetaceans that have convergently evolved to large brains with primates is different to the pattern you see in primates. And actually, when you look at the most encephalized species like the dolphin, you can plot changes in body mass, brain mass in red, and EQ in green. What you find is that the major increases in encephalization quotient occur where body mass decreases. And these are just other plots to show some other species. This one's quite interesting because this is the sperm whale that has one of the largest brains to ever evolve. This is the blue whale. And you can see that they have this nice progressive trend to expand, but in the toothed whales that are notoriously clever, there's less of a directional pattern of evolution. And we've also been looking at whether or not you can see signatures of positive selection on these microcephaly genes in other mammals. This time looking at some more genes that were identified that are associated with the disorder since we last studied them. But here we're looking for positive selection in primates, in non-primates, and then across the combined data. This is the phylogeny of the species involved. These are the primates. We've got a bunch of rodents and rabbits and things. Down here are the ungulates and a dolphin and some insectivores, some Aphotheria. And basically the pattern we observe is that there's positive selection across primates that we knew. There's also positive selection across everything else. So this suggests that whatever is going on in primates might also be going on in other mammals. So there's nothing special about the gene phenotype observations that we found in primates. We tried to test this on looking at these two subclades where there's the most phenotypic data. And again, for ASPM in red and CDK5-AT2 in blue, the two genes that we found an association between DNDS and brain mass, we find the same sort of phenotypic correlations in these two subgroups of non-primate mammals. So this suggests tentatively that maybe the sort of gene-phenotype relationship we observe in primates is conserved across all mammals, and that maybe these genes that are involved in primate brain evolution, including human evolution, are also involved in non-primate mammal evolution. I should say that two genes, Microcephaly-1 and ASPM, have been looked at in cetaceans in more detail across larger data sets. And again, you see these patterns of positive selection, but this time we don't find any statistical evidence for a gene phenotype coevolution. And this is potentially because of those differences that I showed you in the way in which brain and body mass have evolved in cetaceans. So in cetaceans you see a much higher rate of phenotypic reversal in brain mass. So you have lots of episodes in tooth whales in particular where brain mass has decreased and then gone back up and decreased. And this introduces a lot of noise between potential targets of selection and the phenotypic outcome of that selection. But that's more or less all I was going to talk about. I thought I'd finish with some future directions in case anyone else is interested in where this sort of stuff is going. At the moment, there's ever-increasing amounts of genetic data being produced. Every few months, another genome is sort of sequenced and appears online. But one of the major limitations from an evolutionary point of view is that there's often quite a mismarriage between the genetic data that's produced and the phenotypic data that already exists. And if you're studying something like brain evolution, or if you're studying a group like primates where you don't want to go out and shoot them all, then really you need to be quite concentrated about collecting genetic data so that it corresponds to the phenotypic data that exists. There are also sort of ongoing attempts to improve methods of testing for gene phenotype correlations. I'm not really going to go into that, but the sort of challenges that you face are these reversals in the phenotypic evolution. Examples where a gene might be under positive selection for a phenotypic change, but then on another part of the tree you have a different gene being targeted to produce the same phenotypic change. And that sort of noise that's introduced from sort of normal evolutionary variability. And then one of the big questions is how much you believe these gene phenotype correlations and whether or not we can test them functionally. So there are some efforts going on to humanize mice in particular. So you take a human gene, knock it out of a mouse and stick in the human gene to see whether or not it produces a phenotypic change. So this has been done, for example, with FOXP2, this gene that's been implicated in speech. And when you do that, the mice have various funny things. They squeak in a different way. They're also able to learn mazes differently and various other things. And that's quite nice. But that might be a rarity because that gene is quite conserved and it's only one amino acid that's changed, or two amino acids that have changed in the human lineage. They've also done this with ASPM. So taken a mouse and put in the human copy of ASPM to see if it's produced a human-sized brain, and it doesn't. And that's not particularly surprising because ASPM is a very variable gene. If you look across different species, it's not very well conserved, and it's also functioning in a network of other proteins. So if you change one part of that network of proteins, and it's going to have knock-on effects throughout the network. So just blindly putting in one single human gene into a mouse probably isn't going to have a particularly big effect. More recently, people have been working on these organoids, which are basically human stem cells that have been used to develop organs in a petri dish, and then you can mess around with the genetics and see whether or not a gene has a particular phenotypic change. But this is another really challenging aspect of this work. And finally, I think one of the more interesting things is moving away from sort of gross measures of brain size, like brain mass or EQ, and starting to ask more sort of intricate questions about particular phenotypes in the brain, like neuron number or even cortical neuron number or whatever, and also questions about how different brain regions are related to each other. So for example, it's well documented that if you have selection for increased cortical size, you also get an increased cerebellum size. And there's this debate about whether or not this is because the two are genetically linked, so if you sort of pull on one and the other one comes along automatically, or because they're sort of co-evolving to maintain some function. So I think that's the sort of question that this work is going to sort of move into in the near future. But that's all I wanted to say. So I just thank these people who work on the primate stuff with me, and these people that work on the cetaceans, and these funding bodies, and Kit and Caroline for inviting me. And that's it.
Great, Steve. Thanks.